Hi, Shri here. Let's talk today about how we see things in the cell, an area that's going through a revolution right now. Recently, 2 papers were released on bioarchives, which actually showed us the structure of a protein down to the resolution of a single atom using cryo-electron microscopy. Now, let's break that down. Cryo, because freezing the sample can slow down vibrating molecules. Electron, because you shoot a beam of electrons which bounce back. And microscopy, because you're looking at really small things. Now, this technique, with a few major improvements, better beam shooter, better detectors, lets us now see a lot more things in the cell a lot more clearly. Things like water molecules folded into receptors on brain cells, or proteins that carry iron around the body. Basically, really small things that are a really big deal. And that's the cutting edge.